thank you all for coming to join us in this workshop. And we are going to move right now to the first invited talk of the day from Iain Couzin on the geometry of decision-making. So I will give Iain permission to share his screen, I think, and we will move on to his talk. So Iain is the director of the Max Planck Institute of Animal Behavior, the Department of Collective Behavior, and the chair of Biodiversity and Collective Behavior at the University of Konstanz in Germany. His work aims to reveal the fundamental principles underlying evolved collective behavior, and his research involves the study of a wide range of organisms, including insect swarms, fish schools, and primate groups. So Iain, if you'd like to take it away.
Thank you very much. Let's see if this works. Actually, I should just optimize the sharing for video so you can see some cool videos. Okay. I can't see the chat or anything like that. Is that looking okay?
Yeah, that looks good.
Yep,
people feel free to enter questions in the chat. There will be 15 minutes of talk followed by 10 minutes of questions. So during that time, feel free to either speak up or enter your questions and we'll ask them for you.
So yeah, so I'm going to talk about collective information processing. And so for many years, I've been studying these sorts of groups. So this is a fish school being attacked by a predator. And when we look at collective behavior in the natural world, such as this, there really is a notion of information. That's what differentiates collective behavior in biological systems from physical systems, for example. And as was mentioned in the introduction, there's really been a revolution in our understanding of how to get data from such complex systems. So here is a free and open source software package that we've developed here in Konstanz that allows you to track large numbers of individuals while automatically identifying who is whom. And this works much, much faster and with way less of a memory footprint than any previous solution. You can also calculate, for example, the visual fields, which turns out to be very important for many species. It's also independent of species. You can work with rodents or with insects and so on. And this really gives us the opportunity to ask very quantitative questions about behavior. So really, and also we've got sort of data visualization and analytic tools built in. So the idea is to make it very user-friendly and very easy for people to understand. And what I'm fascinated about is cognition. We often think of the brain as the seat of cognition. But when we look at collectives, individuals are actually embedded within this network of communication. And so is there such a thing as a collective mind within groups? And what we've been able to do using these machine learning techniques and actually reconstructing the visual fields and working out which features individuals pay attention to when changing behavior is reconstruct these invisible networks of interactions within groups. If you're a neuroscientist, you can go into the brain to look at the physical and functional connectivity among the neurons. But when you're looking at collective behavior, the interactions among the individuals have no physical form. So we need to use these new technologies to reveal them and study them. And we've been able to show that computation doesn't just occur in the brain, it also occurs in this network. And this occurs in a wide range of scenarios. For example, sensing long-range environmental gradients. The individuals are not capable of even knowing there's a gradient, yet the collective is. In terms of regulating social contagion, the spreading of behavior, and also how groups modify signal to noise ratio is not going on in the brain. They don't change the algorithm in the brain. What they do is they change the structure of the network and let the network perform the computation for them. And that might sound a bit group selection-y, but we've also shown that these properties, these collective properties, readily evolve among entirely genetically selfish individuals. In fact, most organisms we study, there's very low relatedness. And this is much more reminiscent of what neuroscientists have been studying for many decades, is the computational properties that arise not just from the computation within the neurons, but from the network of communication among the neurons. And so when I published this paper in 2007, I really more or less thought it was a metaphor, this connection between neurobiology and what's going on in the individual brain and what's going on in animal collectives. I was very cautious about drawing direct analogies between the two. But today I'm going to go beyond the metaphor. Now I think there actually are genuine connections revealed through mathematics that allow us to connect representation at these different scales of biological organization. So in today's talk, I'm going to discuss some unpublished work. Actually, it's on the archive. How do neural collectors, how do individual brains make decisions? I'm going to take a perspective that really came from studying animal collectives for so many years. I'm specifically going to talk about animals moving in space and how they make spatial decisions, whether they're invertebrates or vertebrates. For example, there's a lot known, some really beautiful work, especially at Janelia, looking at the ellipsoid body in flies and how they integrate vectorial information. So neurobiologists have really found or beginning to find these vector integration circuits in the brain of both invertebrates and vertebrates. So this is really exciting because I've been studying vector integration, the animals themselves being vectors or having internal vectors. And I've become really fascinated by whether the algorithms that we found at the collective scale also apply to within the brain, the neural collective. And so I'm going to focus on vectorial representation in the brain of both insects and vertebrates. To study this, it turned out we needed to develop a new technology. It's very difficult to do these types of experiments. So we developed what we call immersive or holographic virtual reality for freely moving animals. These animals are so small, our fish are less than 1 centimeter long, we can't put headgear on them for obvious reasons. And it uses an old illusion called an anamorphic illusion. And it's so strong that I can tell you right now that this tape does not have a volume above the table. It's just the perspective is that makes your brain believe it to have three-dimensional form. And even when the picture of the tape is rotated into roughly the right position, your brain will pop it back into 3D. There's nothing you can do about it. Similarly, this shoe is not actually three-dimensional, but your brain believes it to be. And so we can use this illusion by tracking the motion of the animals. We do this at 100 hertz. The animals can then move behind and then fully interact with virtually projected objects. And in the context of social behavior, I'm going to look at both social behavior and non-social behavior today. We can create photorealistic, naturally body undulating virtual conspecifics. And using this illusion at 100 hertz, we track the animal in 3D. We reconstruct the perspective exactly from its viewpoint. You can see that the real fish with the red trail believes the virtual fish to be in the tank with it and follows it, even though the projection surface is only the surface of the bowl. Now, one constraint here is, these are our virtual reality systems, is that if I put two individuals in this near arena, then the world will look distorted to one of them because you have to precisely project it onto the retina of the individual. So we can't put two individuals in each virtual environment, but what we can do is make multiple virtual environments and then network them together such the individual in this near arena interacts in real time with a hologram of the individual in the second and the third arena and vice versa. So here you're seeing two zebrafish interacting not in the same physical world, but in the same holographic world. And for, in reminiscence of the movie, we call this the Matrix. And we've done very careful analysis. We also have a 3D tracking system of real fish. And if you compare real fish interactions between two real fish interacting in the Matrix, it's indistinguishable. In fact, the Matrix data here is slightly better because the tracking is better because we don't have occlusions when the individuals come too close. But identical interactions in these environments. So a powerful tool for studying how the brain makes decisions. And here you're seeing 4 individuals interacting within the Matrix. We now have 6 of these systems. And of course, we can have virtual individuals in here. We can do sort of closed loop experiments, a bit like a dynamical social patch clamp, where we can test our theories in situ with real animals. But because I've become increasingly interested in the brain, I've also turned to studying Drosophila, which is where those beautiful ellipsoid body images from Janelia came from earlier on. And also we're studying plague-forming locusts. We had some of the worst plagues over the last 2 years, the worst plagues in almost 100 years in Africa, exacerbated by COVID and the inability to move products and people around Africa. So we also use virtual reality to study these animals. So the advantages are that we've got a very high degree of control. And the fish we work on are so tiny that the body is dominated by viscous flow. And so they can't use the lateral line or olfaction to mediate behavior, they just use vision. These are larval zebrafish. We can quantify inter-individual differences And we can construct experiments and run through parameter space in ways that is impossible in the natural sort of conventional experimentation. As I mentioned before, what I'm going to discuss today is vectorial representation in the brain, and predictions that we can make about how this should operate. If you're interested, this paper is available in the archive. It's undergoing, we're doing quite a lot of changes now in revision, so it's going to change quite a bit, but nonetheless, the paper is out there for everyone to take a look at. And of course, I would really welcome any feedback you may have, because we do have this opportunity, of course, to revise. So whereas, I've often thought of collectives, moving through space and individuals having desired directions, for example, to two static targets shown in blue and red, here I'm now thinking of neural groups that have preferences for these different targets in space. And due to the time constraints, I'm not going to go through the model now, but we develop a very, very simple model. It's slightly different, importantly, slightly different from the classic ring attractor models used in neurobiology. We have an extra added nonlinearity that was inspired by collective behavior of animals that turns out to be critically important. But more or less, you could think of it as having reinforcement among neural ensembles that have similar preferences towards the targets. If they see a target, they can fire more strongly if it's in the direction in which they're encoding. And we also either can have global inhibition for the whole network or negative feedback, i.e. neurons or neural groups that disagree with each other inhibit each other. It doesn't matter in terms of the results we show. It's a very simple model. But it comes up with some important testable predictions. For example, if we look at an animal such as a fly or a locust moving towards two static objects, two pillars in space, we have this reflex behavior to move towards them, to land on them, for example. Then as the animal is far away from these targets, the angle between them, which is shown as theta, will be small. And the model predicts that as they approach the targets and this angle theta increases as they get closer and closer, the system, irrespective of neural noise, will undergo what's called a phase transition, a sudden transition whereby the neurons average the inputs and they choose the average direction, even though no neuron knows the average direction, to suddenly choosing one or other option. And this is a kind of interesting hypothesis, because what it predicts is that as an animal moves towards two static options, it will reach a critical point at which it will suddenly choose one or the other. And I couldn't find any data from any animal where people have conducted experiments and actually tracked the motion of individuals as they move through space when decision making. It's often considered that motion is the output of decisions, but here the motion is integral to the decision making. There's a recursive feedback between the inputs that define the movement, which determines what the inputs are, determines the movement, and so on. And so we should see this behaviorally. We should also see it in the brain, but we should see this behaviorally. Another important point to note is if a system is undergoing a phase transition or a phase-like transition, every single system that undergoes a phase transition, be it physical or biological, has an increase in what's called susceptibility near the critical point. And this is the sensitivity to the inputs. It's an emergent property of the collective system. And that means the brain spontaneously at the critical point will become very sensitive to any slight differences between the options without us having to put that in. It's a wonderful property for a decision-making system to have. And so we were excited about this because in principle, we can test this, right? We should see the animals as they move through space exhibiting this behavior. And as I said, there's been no experiments to my knowledge that studied this, even though there's many decision-making experiments. And so what you're seeing is a heat map. So because we have hundreds of experiments, it's hard to work out what the mean behavior is of the flies in this instance. And here you can see the heat map showing this bifurcation in both the flies and in the locusts. What about three options? Neurobiologists and psychologists often deal with two options. In fact, they often put the animal in between the two options, which is a pretty bad thing to do, as I'll show you in a moment. And what's really cool about the model is, and I wouldn't have predicted this, is that it predicts that the brain spontaneously, and we don't add this into the model, there's no extra features. The brain spontaneously breaks the world down into a series of binary decisions. So the brain deals with spatial complexity by breaking the world up into a series of binary decisions. And for the reasons I just showed you, close to each critical bifurcation, the emergent property of the system is such that it's extremely sensitive to differences among the options. And indeed, in our experiments with both flies and locusts, we find strong evidence of this double bifurcation as predicted by the theory. What about more than 3 options? Well, in our paper, we showed some experiments, but this is a simulation. It works with any number of options. It also works in 3-dimensional space, incredibly robust. Now, I've looked at 2 insects that are unrelated to each other, but nonetheless, we want to understand if this is a general principle. And so also turning to our virtual zebrafish, we can conduct experiments where we change the angle between 2 virtual fish, and we predict a bifurcation in the brain of the real fish, which is indeed what we find. And with 3 fish, we also predict a double bifurcation. It looks a bit different because now instead of static targets, they're moving targets. And that's exactly what we find in the brain of the real fish. And so now working with Armin Weil, who's just moved from Harvard University from Florian Engert's lab, we're now seeking the neural basis of these vectorial decisions in zebrafish. And this has then inspired us to go back to our computational models of animal collectors, wildly different models, but yet they have the similar macroscopic characteristics of feedback loops. And indeed, we now find that we predict animals should do exactly the same thing. And so, this is reminiscent of some work I did quite a while ago, looking at bifurcations in collectives. And now we can actually test this with real data in the brain of even wild animals like baboons. So all of this work's been done in the Center of the Advanced Study of Collective Behavior in the Max Planck Institute of Animal Behavior, where we have a really interdisciplinary team working on all aspects from cellular collective behavior to human collective behavior. We opened our new building last week, just on Wednesday last week, and it's on the edge of campus overlooking the mountains and the lake. It's a really beautiful place. This is the view from our building. And so after COVID, I would be very happy to host anyone to come to Constance. And thanks very much for listening.
All right, thank you so much, Iain. I've turned on auto transcription just because the CVPR organizers requested that. Hopefully that's not too distracting. But yeah, if we have any questions from the audience or from YouTube.
I have a question, Iain. Thank you for a great talk. And I didn't understand what's the experimental setup to test this multi-decision for the animals. How do you propose different options to them?
Yeah, that's right. So in the case of the insect, in the case of the flies and the locusts, it's very simple. They have a reflex reaction to go towards any vertical object. So give them a vertical cylinder, for example, and they will move towards them. They'll be attracted to that. It's a classic reflex in those animals. And so that's what we use. It's a very simple perceptual decision-making paradigm in the case of the insects. In the case of the fish, we turn to a different ecological context. We look at schooling, where they're attracted to other virtual individuals that are moving through space. So for simplicity, we just assume they move linearly, and we can then change the angle with respect to the local individual by changing the distance. And the model is, and by the way, we use exactly the same model with exactly the same parameters for all of these different systems. The model is so robust that you don't need to tweak it to suit different systems. And so yes, 2 different ecological contexts and 3 different species.
Thank you. I want to have a quick follow-up question. So in the plots you were showing, the setup was highly symmetric, namely the central option was in the middle, along the midline of the visual field. And I was wondering if you tested by moving the midpoint to the left or making asymmetrical in some way, just to verify that we're not testing symmetries instead of real decision qualities of animals.
It's a really great question. Of course, it's symmetrical if the animal is pointing directly at that central target. As the animal deviates from that, it won't be symmetrical in its visual field, depending on where it is in space. But you're absolutely right, of course. And we did do exactly what you suggest. We changed the asymmetry between the targets. And so you have 2 targets closer and 1 target further away. And that's in the supplement of our paper, but also the referees said the same thing. So we've done a ton more experiments with asymmetric targets that we will add to the paper in the next few weeks or month or 2 to show that the model is very resilient to that, but also the animals behave as we predict in the model. So it's not an artifact, as you suggested it could be, of just the symmetry of the scenario. That's a really good point to make. Very robust to asymmetric options.
Thank you. Thank you.
So I had another question. You've talked here about deciding between two options like left and right, but the animal also kind of has a decision of swimming or not swimming. So do you think that there's a similar kind of choice that is choice process that's taking place there when the animal decides to initiate movement or stop movement to drive itself towards this critical point?
Yeah, so that's a really interesting issue that we've also addressed. So in the case of the flies, for technical complexities, we actually assume the average flying speed of the fly. So it's not a brilliant system for asking, stopping, because flies don't sort of stop in space, but the locusts do. The locusts classically move in a very intermittent way. They walk and then they stop, and then they walk and then they stop. And so we asked whether, for example, they stop more at the critical point, to sort of, and so on. And there's no evidence, at least so far, there's no evidence that they do. But of course, the other thing about the flies is the neural dynamic, the timescale of neural dynamics is actually pretty slow compared to this timescale of the motion. They move really fast through space. The locusts move slowly through space. They walk slowly towards 5-meter targets away, or virtual 5-meter targets away. So there they have much more opportunity to stop and go. And so there's no evidence that, for example, they stop. In the case of the fish, they have such a strong propensity to school that they more or less maintain. And also in our paper, we have the density distributions and so on. So you can see they really maintain these positions quite precisely within groups. So I think it depends from species to species, but we are looking at that in more detail, in particular in the case of the locusts, where the time scale of the neural dynamics is much, much faster than the time scale of the decision making. It's the only of the three systems where that's the case, and where we can actually access the neural dynamics in that system. Not nearly to the degree you can with Drosophila for many, many reasons, but nonetheless, that's a place to start. And the other thing to do, of course, if you have closed loop feedback in Drosophila, you could force the animal to go into the critical state and determine from its behavior whether it's in the critical state or not, and then look at the neural dynamics. And we would predict, for example, scale-free, length scale-free sort of correlations. At that, there should be something really special about the ring attractor at that particular point, according to our theory. But because I'm not a neurobiologist, I thought we'd get this paper out and then hopefully collaborate with neurobiologists because we have this testable prediction. And now what we're also doing in part of the revision process is we're now questioning whether the classic Janelia model would actually make this prediction at all. We think it won't. We think it doesn't have, there's one level of non-linearity that came from our inspiration from animals that we put into the model, and we've only just realized is not in the neural models. So now we're going back and doing all of this work again, modeling-wise, with the Janelia model from Shaul Druckmann, and trying to see whether that would also make this prediction. We think it won't. We think actually then it sort of invalidates that model. It necessitates or makes a prediction that the brain must have this added nonlinearity. Where it comes from, we don't know, but we can speculate from. But that's just kind of literally what we're working on right now. And I don't, it's a bit hand wavy. But of course, when we, it'll take us a couple of months, I think, to get to the bottom of that.
Is there time for another question, Anne?
Yeah, we have 2 minutes.
Okay, great. Thanks, Iain, for the talk. So as a bit of background, I do a lot of work in like active learning and active experiment design. So I'm quite impressed with your experimental setup. And I'm wondering, what might be some of the opportunities to, as you say, hallucinate agents, test out more complex hypotheses, maybe test out hypotheses you cannot test exhaustively, so you have to selectively design the experiment in a combinatorial design space. I was wondering if you had any thoughts on that.
This is a brilliant question. I would love to connect with you so we could discuss this properly offline, because that's exactly what I've been thinking as well. Whether we can actually have a closed loop between the results coming in and the experiments. How do we help design the experiments using this closed loop feedback system? We've been thinking about this not only in this virtual reality system, but also in our studies of collective dynamics. Whereby we can have this recursive feedback between the structures that we can 3D print, and then the response of the collectors to those structures, and then that informs the model. So we can sort of learn dynamically, and the theory can then tell us which experiments to conduct and validate, because you may actually want to conduct experiments that seem completely wacky to a human, but it makes a lot of sense to reduce uncertainty for large areas of uncertainty in parameter space. And so that, in the VR stuff I showed, we just brute forced the whole parameter space, but you could do, that's really not possible when we deal with the collective behavior. So we need to be smarter. And so I would love to chat to you about that. I think it's a really cool idea.
Great, love to connect later.
Thank you so much, Iain.